Hello everyone, how's it going? Today I'm going to show you guys Mendelian genetics. It is pretty basic, but it can be hard to understand at first, so I'm going to not show my face in the entire video, and I'm just going to show you images and videos of crested geckos so that you understand it better. First, we're going to go over the most basic genetic concepts. An allele is one of the two letters that code for a gene. In genetics, we use letters to indicate whether a copy of a gene is being passed or not. When a male and a female are crossed, they either pass or not pass a copy of a gene. Phenotype is how the gene in question is expressed, so how it looks. It might be a red base, a black base, a yellow base. The genotype is what the alleles, so they're big A, small a, small a, small a, big A, those are the alleles. So the genotype is what the alleles are for this expression of the gene. So a black base maybe is big A, or big A, small a, because a black base is a dominant mutation. A dominant mutation only needs one copy of the gene to express a trait or morph. This means that it can be big A, small a, or big A, and still express the trait or the morph. So the genotype for a black base can be either big A, small a, or big A, while the phenotype is going to be black. If the genotype was small a, then the phenotype would not be black. A recessive mutation is a mutation that needs two copies of a gene to express the trait or morph. For example, the phantom gene is a recessive mutation, so each of the parents is going to have to pass a copy of the phantom gene, so two copies of the phantom gene total, for the kid to express the phantom mutation. A homozygous individual is an individual that is carrying the same letter twice, for example, big A, or small a. For example, since the phantom gene is carrying two copies, it is going to be considered a homozygous individual. A heterozygous individual is an individual that has different alleles, so big A, or small a, big A. An example of this will be all lily whites. All lily whites are big A, small a because they only need one copy of the gene to be shown, and if it had two copies of the gene, it would die. Therefore, all of the lily whites that are in the hobby are heterozygous for the gene lily white. Incomplete dominance is a type of mutation that has a middle form. Big A is going to be something, big A, small a is going to be a different thing, and small a is a different thing. An example of this is the cappuccino gene. While big A is a super cap that has small nostrils and it is super dark and all those things. While big A, small a is the normal cappuccino. And small a means that it carries no copies, so it is not a cappuccino. A recessive lethal mutation is a mutation where the phenotype big A, meaning two copies of the gene, is dead. So two copies of the gene will kill the offspring. An example of this is the lily white gene. Small a is no lily white because it has no copies. Big A, small a is lily white, and big A would be a dead super lily white. Now that we have covered the most basic genetic concepts, we can start seeing how these mutations pass on. This is a Punnett square, and it is how we cross an animal to another animal and predict the kids that are going to have. It is really useful when we're trying to breed animals, and it is also a very easy and accurate way of seeing how the kids are going to turn out. For the dominant Punnett squares, we are going to use the black base that we know is dominant. When we cross a homozygous dominant to a homozygous dominant for black base, meaning big A to a big A, the only kids that we can produce is big A. This means that if you pair up a black base to a black base and all of the kids through years of breeding are black base, then both of the parents are going to be homozygous dominant. It is also important to know the genotype, so big A or big A, small a, because if you want to pair that same mom and dad to a different mom or a different dad, you can predict more accurately what the babies are going to look like. So if you know that one of the parents is big A or big A, small a, this is going to be helpful when you have to pair it up to a different genotype and you're going to be able to predict the offspring. So the ratio of the kids helps us know what the genotypes of the parents are. Therefore, when you pair up a big A to a big A, all of the kids are going to be big A because the only gamete being passed on is a big A. The phenotypic ratio is going to be 1, and the genotypic ratio is going to be 1 because there is only one kid. When you pair up a homozygous dominant to a homozygous recessive, meaning a big A to a small a, all of the kids are going to be heterozygous. This is because the only gametes that are being passed on are big A and small a, while the parent that carries big A is only going to pass big A The parent that is carrying small a is only going to pass small a. When you put together the gametes, the only possibility is big A small a, and this means that all of the kids are going to be heterozygous for black base. When you pair up a homozygous dominant to a heterozygous, it gets a little bit more tricky because now you have more possibilities for the gametes that are being passed on. One of the parents is passing on the big A, while the other parent is passing on a big A and a small a. Therefore, the possible combinations that we can do between the parents are big A to big A and big A to small a. Therefore, we're going to have two possible genotypes, but one possible phenotype. This means that all of the kids are going to look like a black base. However, half of them are going to have two copies of the gene, while the other half is going to only have one copy of the gene. This means that one half is homozygous dominant while the other half is heterozygous. When you pair up a homozygous recessive to a heterozygous, you're going to be passing on a small a from one of the parents and a big A and a small a from the other parent. This means that one of the kids is going to be heterozygous and the phenotype is going to be a black base, while the other kid is going to be homozygous recessive with a phenotype that is not a black base. So half of the babies will look like a black base and only carry one copy, while the other half of the babies will not look dark and will not have the gene that codes for a black base. Lastly, when you pair up a heterozygous to a heterozygous, both of the parents can give either a big A or a small a. Therefore, it once again gets a little bit trickier because we have 4 possibilities instead of 2. You can either get big A, big A, small A, big A, or small A, small a. While big A, small a, and small a, big A are the same thing, however, since they are repeated, the ratio is going to be bigger. So the genotypic ratio is going to be 1 big A, 2 big A, small a, and 1 small a, small a. While the phenotypic ratio is going to be 3 black base to 1 not black base. Now let's take a look at recessive mutations. For the recessive mutation, we are only going to be focusing on the phantom gene. There are lily whites here just for illustration, but we are not focusing on the lily whites, okay? The only gene that we're focusing here right now is on the phantom gene. In the first slide, there are no lily whites, but you will see them further on. So let's say that we cross a phantom to a phantom. All of the kids are going to be phantom because the only gamete that is being passed on is small a. As we said before, big A is used for dominant traits and small a is used for recessive traits. Since the phantom gene is a recessive mutation, we are using the small a. Therefore, each small a is going to be a copy, while a big A is not going to be a copy of the phantom gene. Now going back to this pairing, a phantom to a phantom is only going to be able to produce phantoms, because the only gamete that is being passed on is a small a from each of the parents, because both of the parents are homozygous recessive. Therefore, all of the kids are going to be homozygous recessive for the phantom gene, which translates to a phenotype that is the visual phantom. When we pair up a phantom to a non-phantom that has no copies of the gene, therefore a homozygous recessive to a homozygous dominant, the gametes being passed on are small a from the phantom and a big A from the non-phantom with no copies of the phantom gene. All of the kids that are being produced from this pairing are going to be heterozygous for phantom, so they are 100% het phantom. If some of the kids produced from a non-phantom to a phantom were phantom, this would mean that the non-phantom parent does carry a copy of the phantom gene. However, if all of the kids turn out non-phantom, they will all carry a copy and the parents will be small a, and big A. When we cross a homozygous recessive to a heterozygous, the gametes being passed on are small a from the phantom and big A small a from the non-phantom that carries a copy. The kids that are going to be produced from here are going to be half not phantom, but carrying a copy, and half phantom. So the phenotypic ratio is 1:1 and the genotypic ratio is 1:1. Now, this pairing visually might look like the same pairing that we said before. However, the non-phantom parents from the last pairing was a homozygous dominant, which means that had no copies of the phantom gene. And the parent from this pairing is a heterozygous, which means that it is carrying a copy of the phantom gene. So this pairing can produce phantoms, while the other one can only produce heterozygous phantoms. So if you're a crested gecko breeder looking to make phantoms, in my opinion, your best way of doing it is pairing a phantom to a phantom, but you can also do it by pairing a phantom to a heterozygous phantom. This is why knowing lineage is really important, and when you ask for a picture of the parents, if you see that one of the parents is a phantom, that means that the crested gecko that you're looking for is going to be heterozygous for the phantom gene. When we pair up a homozygous dominant, which means that there are no copies of the phantom gene, to a heterozygous, which means that there is one copy of the phantom gene, we are going to get half of the kids no phantom with no copies, and half of the kids heterozygous phantom. So visually, all of the kids are going to be not phantom, however, half of the kids are going to have a copy of the phantom gene. So the phenotypic ratio is going to be 1 because all of the kids look the same, but the genotypic ratio is going to be 1 to 1. When we pair up a heterozygous to a heterozygous, it once again gets a little trickier because we have 4 possibilities instead of 2. 25% of the babies will not carry the phantom gene and will not look phantom. Half of the babies will be heterozygous for phantom, and another 25% of the babies is going to be a visual phantom. If you're into reptiles and you bought them before on Morph Market or on other sites, you've probably seen people that say 66% possible het or things like that. This is where this comes from. Out of the 3 kids that do not look phantom, there are 2 kids that are heterozygous for phantom. Now visually, we cannot tell them apart, but 2/3 of the kids are going to be heterozygous, and 1/3 of the kids is not going to have any copies. That's why it is good to indicate that there are a 66% chances of the not visual phantom kid to be a heterozygous phantom. Now let's take a look at incomplete dominant mutations. For this example, I am using the white wall gene. 2 copies of the gene will make a thick wall on the dorsal of a crested gecko, while 1 copy of the gene will make a few dots on the side of the crested gecko. The dots on the side of the crested gecko are called whiteout. So the homozygous dominant big A is a white wall, while the heterozygous is a whiteout, and the homozygous recessive is no copies of the gene and no expression of the gene, which would mean that there are no spots and no white wall. When we cross a white wall, so this is a homozygous dominant, to a no white wall, no whiteout, so a homozygous recessive, we get 100% whiteout kids. The gametes that are being passed on are a big A from the homozygous dominant and a small a from the homozygous recessive. The only possible combination that we can make out of these parents are big A, small a. So all of the kids are going to be whiteout, which is big A, small a, which is heterozygous. When we cross a homozygous dominant to a homozygous dominant, the only gamete being passed on is the big A. So the only possible combination that we can make between these 2 parents is big A, big A. Therefore, all of the kids are going to be white wall kids, which are big A, homozygous dominant. When we cross a whiteout, which is a big A, small a, to a small a, which has no whiteout and no white wall, half of the kids are going to be whiteout, and half of the kids are going to have no copies of the gene. This is because the gametes that are being passed on from the whiteout parents are big A, small a, while the gametes that are being passed on from the no whiteout, no white wall parent is only small a. So the possible combinations that we can make are big A, small a, and small a, small a. Since there are 2 possible combinations, there are only 2 possible genotypes. The genotypes will be big A, small a, and small a, small a, and the phenotypes will be half of them whiteout and half of them no whiteout, no white wall. When we cross a heterozygous, so a whiteout, to a homozygous dominant, so a white wall, we have half of the kids whiteout and half of the kids white wall. This is because the gametes that are being passed on are big A and small a from the whiteout parent and only big A from the white wall parent. Therefore, the possible combinations are big A, and big A, small a. Big A would be the white wall and big A, small a would be the whiteout. Again, since there are only 2 possible combinations, half of the kids are going to be whiteout and half of the kids are going to be white wall. Now again, when we cross heterozygous, we have 4 combinations, so it gets a little bit trickier. When we cross a whiteout to a whiteout, we have 1 out of 4, so a 25% chances of having a white wall. We have 2 out of 4, so a 50% chances of having a whiteout, and 1 out of 4, so a 25% chances of having no copies of the gene, so no whiteout, no white wall. Now let's see how recessive lethal mutations pass on. Recessive lethal mutations work exactly the same than incomplete dominant mutations, but are different because the super form, meaning the two copies, is dead. While one copy of the gene will make a perfectly healthy animal, two copies of the gene will be lethal to the kid. Therefore, when we have a recessive lethal mutation, we avoid breeding two animals with this mutation to avoid having a fourth of the kids die. Since the homozygous dominant form is dead, we do not have a lot of possibilities of what to breed to our heterozygous form. Therefore, I only prepare two slides for the recessive lethal mutation. When we breed a heterozygous, meaning that it carries and shows the mutation, to a homozygous recessive, meaning that it does not carry the mutation, we're going to have half of the kids with the mutation and half of the kids without the mutation. So big A small a is going to be our lily whites, and small a are going to be not lily white. Many people have asked me, so I have a crested gecko that has a lily white parent but is not a lily white. How many lily white kids can I make out of my non-lily white kid? The answer is 0. If you have a non-lily white crested gecko, there is no way on earth that you're gonna be able to make a lily white crested gecko. This is because lily white is not recessive, it is recessive lethal. This means that you need one copy of the gene to be expressed, not two. Two would kill the crested gecko. When we breed a heterozygous to a heterozygous, meaning a lily white to a lily white, half of the babies are going to be lily white, 1/4 of the babies are going to be dead, and another fourth of the babies are going to be non-lily white. Also, in this comparison, you can see if you breed a lily white to a non-lily white, you're going to have 50% kids lily white, and if you breed a lily white to a lily white, you're also going to have 50% kids lily white. Therefore, it's always going to be better to breed a lily white to a non-lily white than a lily white to a lily white, because you get half of the babies lily white, and if you're breeding a lily white to a lily white, 1/4 of the babies is going to be dead. Lastly, I want to show you guys how to do pairings with multiple genes. So we're not just focusing on the lily white gene, but we're also focusing on the red gene now. I'm not 100% sure on this. Some people say that the red base is recessive, but I call it incomplete dominant, because there is thought to be a blush on the animals that carry one copy of it. Therefore, they are expressing something about that gene. So the illustrations that I'm going to do are based on the fact that red base is incomplete dominant instead of just recessive. The blush that I'm talking about is a red chin, or also called red neck. It is thought to happen on individuals that have big A small a, and the individuals that have big A are going to be fully red. Since we have more than one mutation, we're going to use letters that indicate what mutation we're talking about. So instead of using big A, we're going to use big R for the red mutation, and instead of using big A, we're going to use big L for the lily white mutation. Now, let's pair up a red not lily to a heterozygous red lily, therefore a lily white that has a blush on the neck. The parents are going to be big R, because as we said, a red base is two copies of the gene. Therefore, we're going to pair big R, small l, to a big R, big L, small l. As we said before, all lily whites are big L, small l, because if they were other thing, it would be not lily white or a dead baby. Therefore, the gametes that are being passed on are big R, small l from the red base non-lily white parent, and big R, small r, big L, small r, small l from the lily white that has a blush. We can do 4 possible combinations. So we're going to put the gametes of one of the parents as rows and the gametes of the other parent as columns, and then we pair them up and see the possible combinations. 1/4 of the babies is going to be big R, big L, small l, which is going to be a red lily white. Another 1/4 of the babies is going to be big R, small l, so it's going to be a red not lily white. Another 1/4 of the babies is going to be big R, big L, small l, which is going to be a het red, so a blush lily white. And the last 1/4 of the babies is going to be heterozygous for red, so a blush, but not lily white. When we pair a heterozygous red not lily white to a heterozygous red lily white, we have even more combinations. The gametes for the heterozygous red non-lily white are going to be big R, small l, and the gametes from the heterozygous red lily white parent are going to be big R, small r, big L, small r, small l. So we're going to put the gametes of one of the parents as rows and the gametes from the other parents as columns, just like we did before. The possible combinations are big R, big L, small l, which is going to be a red lily white, big R, small l, which is going to be a red non-lily white, big R, small l, which is going to be a heterozygous red lily white, big R, small l, which is going to be a heterozygous red not lily white, big R, big L, which is going to be another heterozygous red lily white, big R, small l, which is going to be another het red not lily white, small r, big L, which is going to be a no red, so no het, no copies, lily white, and then a small r, small l, which is going to be not red, not lily white, so it's going to carry none of the genes. Now let's do it even harder. We're going to do a pairing between two fully heterozygous parents. I'm not going to go over all of the kids because I don't want to be reading big R, small l, and all those things, but you can make yourself an idea and appreciate the time that I spent on this slide. Also, since in this pairing we're breeding a lily white to a lily white, note that 1/4 of the kids, so 4 out of 16, are dying. And lastly, I want to say that there's no limits. You can do as many genes as you want, but it gets way harder with Punnett scores. There are other methods, but with Punnett scores, I wouldn't do more than 3 genes. It gets really hard, especially if it's all heterozygous to a heterozygous, but this is an illustration of what a heterozygous with 3 genes to a heterozygous with 3 genes would do. So the pairing here is going to be a heterozygous red, lily white, heterozygous Dalmatian to a heterozygous red, lily white, heterozygous Dalmatian. And those are all of the possible kids. There's going to be several of one kind, several of another kind. There's going to be several phenotypes that are the same, several genotypes that are the same. So if you want to do that, you can do it on your free time. But honestly, I spent too much time on this, and I don't think that reading it out loud would even help you that much. But anyways, this is all I have today for the illustrations. I hope this was useful. If it was, please check the next videos that I'm going to be doing on different traits and genes and how to make them express in crested geckos, how to pass them on, and how to breed for those specific genes. Thanks for watching, and I'll see you in the next video. Bye.